Chapter 28, Classification. Pages E1. This is based on the classification on pages E1 and E2. And that's mainly what I'm going to use is the classification from E1 and E2 throughout the book. Okay, whenever for the rest of the semester we'll be using mainly that classification. So let's just go through some general information. Many years ago, we used to have a kingdom called Protista. Remember the 5 kingdoms? Well, one of them was Protista. It basically was any organism that was single cell, that wasn't really an animal, wasn't really a plant, wasn't a fungus, wasn't prokaryotic, so we just threw it in here in the kingdom Protista. It was one of those catch-all, made no sense kingdoms. It's a lot easier to remember, actually, but polyphyletic. Just, it was not monophyletic in any way, shape, or form. So biology uses DNA technology to look at how organisms are related. We produce monophyletic trees, right? That's what they're trying to do, is they're constantly trying to produce monophyletic trees, which are what?
Clades.
Clades. They contain clades. And a clade is what? A common ancestor and all of its descendants. So it's really, really important that you get these things in the correct area. Now, one of the things about science is you can come up with this hypothesis, which is what a monophyletic tree is a hypothesis, and then you test it. If it's not supported, what do you have to do in science? Change it, right? Okay, so that's why this is constantly changing. Over and over, it constantly changes. So right now this works. I'm sure in the new edition it will be very different. So we're going to go through Protists. The kingdom Protista no longer exists. It has been divided up into many different kingdoms. Still in the domain Eukarya, but many different kingdoms. So let's look at some general information. The cell, they're eukaryotic. Most of them are unicellular. The interesting thing is most eukaryotic cells, most eukaryotes, organisms, are protists. So it's not like they're not important organisms. Most eukaryotes are protists, if you take all the organisms. All right, so let's look at some structural and functional material regarding the cell, and just in general. If you're a unicellular organism, which means you're made up of one cell, but you need to do all of life's processes by yourself, which means you rely on the materials inside your body to extract energy, to get rid of waste, to replicate, whatever it might be that needs to grow, to respond. Unicellular organisms are very complex because they need to do all of life's processes by themselves. So they're incredibly complex. They actually, some of them have organelles that other cells do not, like a contractile vacuole. We'll talk about what a contractile vacuole is in just a little bit. Okay. So nutrition. They gain energy and materials in different ways. We have some that are autotrophic. And these are photoautotrophic. Heterotrophic. So photoautotrophic means they use sunlight to produce their own food. Heterotrophic means that they consume other organisms to get their nutrients and their energy. And then we have what we call mixotrophs. They do both. So they can be photosynthetic autotrophs. They produce their own food. They eat something else, or they can do both. They make their own food and they eat other things. I pulled this out of page 252. because we didn't really go through sexual reproduction. There are different types of life cycles. We can talk about something called asexual. Asexual life cycle, when an organism has an asexual reproduction, that means that organism produces offspring by itself, typically by mitosis or binary fission. Sexual reproduction, we get meiosis, so we end up having gametes. And those gametes are fertilized to get a zygote. Syngamy, anybody know what syn means? S-Y-N. Syn. Together. And gamete refers to gametes. Kind of another term for fertilization. It means putting two gametes together. Think sperm, egg. Not quite like that, but you know. All right, so there are different things that we can look at. We talked about haploid a little bit before. I told you if you had problems, come in and ask me. Nobody has come in to ask me about mitosis or meiosis, so I'm just going to go with the assumption you guys understand it completely. Okay, because nobody's come in and asked me, so I have to go with that idea. All right, so haploid, half the number of chromosomes. Diploid, 2 sets. All right? All right, so one of the sexual, these three things here are sexual life cycles. So where you have two organisms. All right, so we can have animals. Diploid organism, that's what the 2N refers to. Cells within that organism go through meiosis, which means you reduce it from diploid to haploid. Gametes are haploid. They come together. That's fertilization. Fertilization is where the gametes come together. You produce a zygote. Okay, it's the first cell after fertilization. It grows up into a diploid organism, multicellular diploid organism. Any questions? Okay, we're not going to worry about that one right now. That's when we get to animals. Plants. In plants, we have something called alternation of generations. Have you guys done that in lab? Not yet? Okay. So alternation of generations, this is where you have two organisms. They're multicellular, so two multicellular organisms that alternate. Okay, so just real quickly, you have a sporophyte. This is a diploid organism. It's the tree out there is a sporophyte. Okay, so if you look out there, that tree right out our window is a sporophyte. It produces spores, which are haploid. How do you go from a diploid to a haploid? Meiosis. The spores grow up into another type of individual called a gametophyte. Haploid. On that tree, there are gametophytes, other individuals. They depend on that tree for nutrients, material, and protection. Sometimes that's not the case. You guys know your mosses? You guys start mosses? I know you did because Dr. Kovnat said you did. Yeah? What's the dominant life form in a moss? Gametophyte. So you know the furry little moss thing that you guys see, that plant? That's a gametophyte. A little tiny stalk that grows out of that plant. So here you've got a moss. Right? Got that little stalk that grows out. Did you guys see that? I don't know if it was in your moss. That little stalk that's stuck up. Usually we have those. Or whatever it is, that's a sporophyte. And that's a different individual. Moths produce gametes by mitosis. The gametes come together in fertilization to get a zygote, which is diploid, and it grows back into a different diploid organism. Alternation of generations. You alternate between multicellular organisms. Multicellular, multicellular. Sporophytes do not produce a sporophyte. This plant does not produce this plant. This plant is a gametophyte. It produces gametes, maybe from another plant also. They're going to fuse. And you get a sporophyte. So alternation of generations. What's a generation? Parent, offspring, right? Those are generations, so that's what that means. You alternate the forms between generations. One generation is a sporophyte. It produces offspring gametophytes that grow up that produce gametes, so the next generation is a sporophyte, and you alternate, sporophyte, gametophyte. These are multicellular organisms. So if you look at that diagram right there, that's what it's showing you. It's showing multicellular organisms. This is, this is a moss. This little thing. So what it's showing you is this sporophyte here, this thing here, is 2N. Goes through meiosis, produces spores, you get a gametophyte. The gametophyte produces gametes, it grows into a different sporophyte. The sporophyte produces spores, grows up into a gametophyte. Okay? It alternates generations. Does anybody have any questions? Because when I get to protists, I'm going to be doing this. When I get to plants, I'm going to be doing a lot. Yeah.
It produced with itself, so it was self-replicating. Would it use its own gametes to
Yes.
fertilize?
Mm-hmm. Without a doubt. Yeah, you mean so sperm, egg get together, doesn't matter. If you've got that sperm, egg get together, you've got a zygote, that zygote produces a sporophyte. Yeah.
So the same process itself to produce both sperm and egg.
Yeah, it would be a plant that produces both types, yeah. Or an organism that produces both types, yeah.
So let's say like the parent is the sporophyte, and then the offspring would be the gametophyte, and then if the gametophyte's the parent of the sporophyte.
Mm-hmm. Exactly. And it's not if. That's exactly what happens. You have a sporophyte generation, so here you have the sporophyte generation. It produces spores. Spores grow up into gametophytes. Gametophytes produce gametes. They fuse, grow into sporophytes, and you just keep going. The thing is, our sporophytes and gametophytes are multicellular. They're actual multicellular organisms. Now, this is not really what protists do, per se, but that's what this middle one's showing you. There's alternation of generations, which is important for plants. All right. Some protists go through a haploid unicellular organism. Yeah.
So what's the gametophytes versus the spores? Like what's the change between the spore stage and the gametophyte?
The spores are produced by sporophytes. Okay, they're actually produced by the sporophyte, the individual, but they grow into a different type of individual called the gametophyte. They're haploid. Yeah. So spores are produced by the sporophyte, through meiosis, which is the production of genetic material. Okay? So you have a sporophyte, they produce spores by meiosis. Those spores do not grow back into a sporophyte. I mean, that's what you would expect, right? In humans, mom produces egg, dad produces sperm. Hopefully you get a human, right? That's not happening. You're getting the same species. Hopefully the human looks like mom and dad, right? Okay. You have the same genes. Okay, so spore grows up into something different. This can look completely different to the sporophyte. You're alternating types of organisms, even though it's the same species. Moss. Actually, the sporophyte on the moss is dependent on that little mossy portion, the little plant that you guys actually look at. That's the gametophyte. That sporophyte is dependent on that. It's short-lived. It doesn't live for a very long time. Once it produces spores, it dies. Not needed anymore. This is the dominant generation, is that gametophyte. We'll get into this a lot more during plants. And no, this is not on the exam, but you should keep it in mind. And the reason you should keep it in mind is you're going to hear it continuously. So when we're talking about animals, we're talking about a different type of sexual reproduction. When you're talking about plants, you're talking about something a little bit different. You kind of got to keep that in mind because I'm going to get to protists, and they are different. And you got to know the life cycles. You have to know. You have to be able to draw them out, which means you have to practice them. And we're going to have something like, no, not these. These are just generalizations. You're going to have to draw them out. That might be your essay. Okay? And I think in here I've got 6, 5, something like that. I don't remember how many I did. All right? So you have to practice. So this is something that's important. This specifically will not be on this exam, but keep it in mind for next time because we circle life cycles. When you do a cycle, you start and end. Okay, that's what a cycle is. All right, so that's what we're doing here. Okay? But not now. All right, so let's do endosymbiosis. All right, endosymbiosis. This is when one cell consumes another cell but the cell that's consumed is not eaten. It's not digested and taken apart. For some reason, somehow, that cell eventually becomes an organelle. So this cell stays in there and becomes an organelle. Quite a bit later. But think about it. What's symbiosis? Symbiosis is a relationship between two organisms. What does endo mean? Inside, right? So it's a relationship between two organisms inside one of them. Okay, the difference is that they eventually, the cell eventually is no longer a symbiote. It's not its own cell. It can't live without. It can't live without whatever cell it's in. 2 examples, mitochondria and plastids. 2 perfect examples of endosymbiosis. Mitochondria. It's been hypothesized that mitochondria are the results of an endosymbiosis with an alpha between a heterotrophic protist. Heterotrophic consumes something else, makes sense. Protist, okay, so eukaryotic cell, and an alpha protobacteria. A plastid is, for example, a type of plastid would be a chloroplast. What they think the endosymbiosis of a plastid is that The first thing that was to develop was an endosymbiosis of mitochondria. Why? Why would that be a logical hypothesis? Why? I know you guys know it. Okay, well, what do all eukaryotic cells have? Mitochondria. So does it make sense that mitochondria would have come first? Do all cells have chloroplasts? Not unless you go out in the sun and produce your own food. No, not all cells have chloroplasts. So it makes sense that mitochondria would have come first, right? Because if all cells have mitochondria, remember, you want less evolutionary events, right? Okay, so plastids, the same kind of idea. What they think happened is a secondary endosymbiosis with a cyanobacterium. What's secondary mean? Yeah, it happens second. That's it. Okay, so this is in your book. I'm not going to go over it a whole bunch because I am running out of time. But you need to know it. So make sure you read through your book. That's basically what I said. You had a heterotroph eukaryote. Single-cell heterotroph eukaryote. Okay? Cyanobacterium. Now this heterotroph eukaryote would have already had its mitochondria. Okay? It's already got mitochondria. When we're talking about secondary endosymbiosis in protists themselves, okay, and specifically protists, we're talking about how different groups of protists got chloroplasts or plastids. And so that's what happened here. What they think is there was a heterotrophic eukaryote that took in a cyanobacterium. Okay? And then you can count up the membranes. I don't care if you do that or not. But basically, this plastid, this here, would have been a symbiote. Over time, it benefited this organism. What does this organism look like? Back in your notes. Okay, so this organism would have been the common ancestor of red algae and green algae. And what they notice is that it's independent. So the plastids in red and green algae actually came about independently. So how would this have happened? Here you have a common ancestor. It would have branched into a red and green algae of some sort, not the ones that we have commonly today. Okay? Both red and green algae do photosynthesis. So what it's hypothesized is that they consumed a cyanobacterium, secondary, another one, that eventually formed different types of plastids in the cells. That's all it is. Endosymbiosis is an organism, a single-celled organism, taking in another single-celled organism. Something happens. You don't get digestion. That organism's not consumed. And then it just continues because it benefits the host. If you benefit that host, then the likelihood is those organisms are going to produce more individuals for the next generation. Yeah? Does that make sense? Yeah, it's all evolution. You guys need to go back over evolution. Right? Because that's all it is. If you benefit the host cell, that organism is more likely to produce more individuals. That organism produces more individuals. Its offspring will have the characteristics that benefited that original host. What would be one of the benefits to that original host? Think about it. What is it doing? What's the benefit? Not free energy, but they're producing their own food. Yes, so they're producing their own food. So if there's a reduction in food materials, this organism doesn't necessarily have to consume. So here you're a heterotrophic, and over time, they diverge into something that is autotrophic. They're able to produce their own food. How did they come up with this? What's the evidence for this? I mean, you say this and people are like, well, that sounds a little far-fetched. What's the evidence? Mitochondria and chloroplasts have their own DNA. They replicate themselves. And guess what their DNA looks like? Bacteria. It's a circular bacterial DNA. So that's a very big piece of evidence to support it. All right, I'm not going to go through all that. You can go ahead and read this. Make sure you read it in your book. All right. So any questions about that? Yes?
Is that how mesothells came about?
How what?
Mesothells came about? Because you said mesothells was one that actual cells were autotrophic and heterotrophic.
I don't know. It's very likely. What they think is that cells that are photosynthetic, so eukaryotic cells that are photosynthetic had to get that somehow. And if you look at plastids, they have their own DNA and their associated bacterial DNA. So yes, it's very likely. But I don't want to say specifically because I don't know that the specifics are out. Okay. This is not in your book. I'm not photocopying any more of these. I did it in color for you. Don't thank me. The only reason why I used my inkjet to do it in color for you is because I forgot to do it in black and white. I get to go buy a whole new inkjet ink. I'm not going to do this for you continuously. Okay? They're on the Elmos. You have to work with them. This, when I was in school, this is the way I did it. Okay? This is what you do. There's a lot of these. I'm not going to lie to you. I'm not going to even sugarcoat it. There are a lot of them. Each time, now, there are some changes I'm going to make. Like I said, it takes a long time to do. As I was looking at it, I realized, oh, I don't like it there. I don't like it there. Right? The best you can do is take this information and learn where things are and how they're related. Are you going to have to draw this tree? Are you going to have to draw this tree? Absolutely not. You will not draw this tree. This tree is for your benefit, and it is strictly for your benefit. Do you have to know these names? Absolutely. Without a doubt, you have to know these names. We have 5 subgroups. We no longer divide into simple kingdom. We go domain and then we divide it out into 5 subgroups. Look, I gave you the 5 subgroups. Excavata. They're in blue. Okay. Chroma alveolata. Rhizaria. Archaeoplastida. Archaeoplastida. Uniconta. Those are 5 subgroups. So in the domain, eukarya, we divide all eukaryotes in and out of this 5 subgroups. You guys all, did you get one? Oh, okay. Divided out in these 5 subgroups. You have to know these subgroups. If you look in your book, you can go off your book if you want. I don't care. But I'm very visual. I have to see things. If I can't see them spatially, I have trouble understanding them. Okay? So that's why I do this. Because it helps me. I know it helped me as a student. This is the only way I could remember anything when it came to biology. Because it is just too much. Okay? To just try to sit down and memorize. All right. So excavata, they have a unique flagellum. Modified mitochondria. Those are general characteristics of that grouping. Do you need to know the general characteristics? Yes, of course. Okay? Chroma alveolata. They have structures called alveola. No, not the same ones we have in our lungs. We don't actually know what they do. We know they have them. We don't know their exact function. Okay? The thing about chroma alveolata is they think that they may have come back by secondary endosymbiosis. Rhizaria, they're put together because they have similar DNA. So the DNA has similarities, that's why they're put together. Archaea plastida, these are the closest relatives to land plants. And Uniconta, these are closest to fungi and animals. All right, now look. This is a clade, right? All of that is a clade. All of that is a clade. They're divided out as clades. All of that is a clade. That is a clade. That is a clade. Sorry. Does everybody see that? Okay. So take a deep breath. You're going to know all of them. But not for the next exam. There are certain things you have to know for the next exam. So let's do it. I'm going to ask you the 5 subgroups. I'm going to ask you their characteristic. I might do it as Mac sheet. I don't know. Haven't written it. No clue. Now, if you look on here, the ones highlighted, you better, I don't think I highlighted yours. After a certain point, I was like, no, you can do it. All right? Highlight everything that I have highlighted. So, Diplomonadia, Parabasala, Euglenozoa. Those are kingdoms. Remember classification scheme? Those are kingdoms. Alveolata, Straminopilia, kingdom. Highlight those. Foraminifera and Radiolera, kingdoms. Highlight those. Rhodophyta, Chlorophyta, Charophyta, kingdoms. Highlight those. The next one is Plantae. It's a kingdom. You don't have to know it this time around. Okay? I didn't put it down. It's Plantae. We'll get to that. There's even more to add there. Amoebozoa, kingdom. Highlight that. And your book divides out Opisthoncons. Yeah, no, it's not a kingdom. They just throw it in there. It's not really a direct. It's a clade. So it's not really a kingdom, but it's part of the clade. Okay? All right. So what are you going to do? What are we going to do? Because I'm going to be out of time real soon. We're going to do, well, everything that says not covered here. Okay? So if you notice, cercozoans, I'm not even going to get to. Okay? I did that for completeness. So that's not going to be covered. Charophyta, I'm not covering them here, so they're not on exam 2. Neither are Plantae, not on exam 2. Nuclearids, not on at all. I'm not covering them. Fungi, well, they'll be on exam too. They're just not right here. Okay? Choanoflagellates, not on exam 2. Animalia, not on exam 2. Okay, so these are all eukaryotes. The next one I have available for you, what will happen is coming off the fungi will be a big list of stuff. Okay? And then the next one will be plantae. Coming off the plantae will be another something like this. I did the work for you. Okay? Because there's nothing more annoying than trying to read that book and figure out what's what. All right? I did not get nearly as far as I needed to, by about 6 pages. I talked about excavata. Okay? So this is what I'm going to do. I'm going to try to keep this flow chart along with the Elmos at the same time. I'm sure I will mix them up at some point. If I do, just yell. Okay? Just let me know. All right. So some general information about excavata. And I actually wrote some of that information for you. Unique flagella. And actually what we call modified mitochondria, which means they're not what you typically think. Okay, so that's the exact same thing that I wrote here. Now there are 2 kingdoms that we talk about, the diplomonads, or diplomonada, and the Parabasala, okay, and the Glenozoa. So you notice I did those in yellow? I highlighted those? Okay, so those are going to be the kingdoms. The first 2 we're going to go over are the Diplomonadia and the Parabasalida. Okay? All right. All right, so if you want to divide these out, I'm going to put general characteristics on the lines. Right? If you look at the back of the book, they give you this table. So I'm going to basically take that table and put it in here for you. So you can define between the different kingdoms and the different organisms. All right? So one of the things that separates this out is they lack plastids. Okay? They also have modified mitochondria, so they've done something to the mitochondria. I'm just going to abbreviate MITOCH, mitochondria. All right, so let's look at the diplomonads. And we have a picture right here, Giardia. Okay, so I'm doing the one on the left. All right, so what are some of the characteristics of the diplomonads? Well, the main characteristic is they have a mitochondria kind of called a mitosome. And mitosomes are reduced, it's up here, reduced mitochondria. They lack an electron transport chain. All right. Now, one of the other characteristics is that they have flagella in a groove. And a lot of them have 2 equal-sized nuclei. They actually have 2 nuclei. So I'm putting main characteristics on here and some other characteristics on there. 2 nuclei. All right. General organism you need to know is Giardia. So that's the organism that you need to be able to identify as diplomonada. Parabasalids have something called a hydrogenosome. They're anaerobic, which means that oxygen poisons them, and they actually release hydrogen gas instead of oxygen. I'm sorry, they release hydrogen gas, and they don't use oxygen. An example would be trichomonas. It actually causes yeast infections. That's one of the things that cause yeast infections in the female vaginal tract. Right? So you need to know trichomonas. Yes?
So are those the is that what being hydrogenosome means?
A hydrogenosome is a reduced mitochondrium. And that reduced mitochondrium, they're basically anaerobic. They're not going to use, it's very reduced. They're not going to use oxygen to break down food material. Okay, put oxygen in poisons them. And so here's your picture, the trichomonas. It's in your book. Okay, so those are some of the excavatas. The other kingdom of excavata are the euglenozoa, right here. Now, euglenas are very diverse. They have organisms that are heterotrophs, autotrophs, mixotrophs. Their distinguishing feature is in their flagellum, which is this here. It's a spiral or crystalline rod that's inside the flagellum. Okay, that's its distinguishing feature. So notice the distinguishing feature to branch out the diplomonadia and the parabasala is going to be that modified mitochondria. The distinguishing feature to branch out the euglenozoa is going to be this rod. They have a rod inside the flagellum. There are two parts to that clade. There are two of them. You guys should have this, so it shouldn't be a problem seeing it. Okay, the kinetoplastids and the euglenophyta. Everybody has this paper, right? Oh, good. Okay, the kinetoplastids and the euglenophyta. The characteristic of the kinetoplastids is something called a kinetoplast. The kinetoplast. Kinetoplast is actually a mass of DNA inside the mitochondria. So it's an actual specialized mass of DNA inside the mitochondria. And if you look at them, okay, now this is, you see little purple ones right here? That's trypanosoma. That's the example. Causes African sleeping sickness. Okay, so it gets in there. It's called African sleeping sickness because it has two stages. The second stage can cause coma. Okay, and it can be fatal if it's not treated. That's the second stage. Notice that they have these little flagella. So they're going to have those rods. The second type is going to be the euglenophytes. An example would be euglena. They're characterized by having two flagella that come out of this enlarged region at one end of the cell. Or one pole of the cell. Now, if you look at the euglena, there are several parts. You really should be able to identify some of these parts. You've probably already done them in lab, right? Done all the parts? Yeah. No? Oh. Okay, that's fine. You might get to it. You might have just wanted me to do it. All right, well, here's the long flagella. Okay, see how long that is? And here's a short one. Do you guys see this little tiny one right here? They come out of the same enlarged region at the end of the euglena. So everybody see that? See the little tiny one right there? And the really large one's that one. If you notice there's this little red portion on it, it's called the eye spot. The eye spot is used, it's actually protective. So what it does is it protects light from getting in to the light detectors. So it protects the light detector by limiting the direction in which light can come in. Light detector does exactly what it sounds like. It detects light. Anybody remember the term used for organism that moves towards light? What is it? Photo. Do you remember the term that means movement, going towards to or away from? Taxis. Yeah, so taxis is movement towards or away from some type of stimulus. These are phototaxis, so they move towards certain intensities of light. Something that you'll see here is something called a pellicle. You see this right here, this to that, this region right there? That's called a pellicle. It's a band of proteins that help keep the shape of the cell. It's protective. It allows for flexibility, it allows for strength and actual movement of the cell. They're found underneath the plasma membrane, okay, so they run the length of the cell. Plasma membrane we've gone over a little bit. It's the outside portion of a cell. There's no cell wall on a euglena. Chloroplasts, photosynthesis. Nucleus contains the DNA. One of the things we haven't talked about is something called a contractile vacuole. And a contractile vacuole, oh, it doesn't show real well here. It'll show better in some of the other pictures. Actually will take excess water inside the cell. It typically has these little radial canals coming out from it. It takes excess water into the contractile vacuole and it ejects it out of the cell. The euglena, the last thing, they live typically in fresh water, so water tends to go into the cell. If too much water goes in, they'll explode. So the contractile vacuole takes that excess water and ejects it out of the cell. Okay? That water typically moves in by way of osmosis. So if you guys don't understand osmosis, make sure you come ask me. I'll go over that real quickly. It's not a big deal. It's just passive movement of water from an area of high concentration to low concentration. Okay. Excavata. Any questions? Oh, okay. Next. Okay. Chrome alveolata. May have originated by secondary endosymbiosis. And if you guys remember secondary endosymbiosis, what happens in primary is a cyanobacterium was consumed by a heterotrophic eukaryote. So we talked about that, right? So the organism is able to do photosynthesis because that's what the cyanobacteria does. And over time, the two organisms become compatible. So initially, they're living in a symbiotic relationship. And then they eventually, the cyanobacterium becomes an organelle. Well, this gives rise to red and green algae. And secondary endosymbiosis is when the red and green algae are consumed by another organism, and so give rise to other types of protists. And these are protists that are believed to have arisen from secondary endosymbiosis. Okay, there are two kingdoms, alveolata and stramenophilia. Alveolata. Alveolata have these membrane-bound organelles right underneath their plasma membrane. We call them alveoli. They're not the same that we think of in our lungs. Okay, so it's under the plasma membrane. We don't actually know what they do. We don't know the function of them. So that's something that's being investigated. Okay, but that's something that actually divides them out. They're called alveoli, so that's what these are right here. And if you look at this picture, that's what these are. You guys should have that on your picture. You guys have that on your thing? Okay, so it's right here. Those are the alveoli. See these membrane-bound organelles? And we're not sure what they do. So, and it completely classifies a group. Nobody finds that neat? I find it kind of interesting that we completely classify an entire clade based on an organelle. We're not quite sure what it does. Okay. All right. Let's go through the alveolata. There are several parts to them, several groups to them. The first one that you're going to look at are the dinoflagellata, or dinoflagellata. All right, well, here's an example. Characteristic, two flagella. See the two flagella? This is one. It goes all the way around. That's one here, okay, inside a groove, and here's a second one inside the groove. So it has two flagella inside a body groove. And the second characteristic is the cell is actually reinforced by plates. So the body is reinforced by plates. I'm sorry? Groove? Oh, that should be, thank you. Physteria is an example. Of a dinoflagellate. It's in you can get it all in your book. Okay, it's a type of dinoflagellate. Partly responsible for things called red tide. Have you guys ever heard of red tide? Yeah, deadly. Can be deadly, so shellfish take these in. Yeah? Okay. All right, second type is apicomplexa. An example of apicomplexa is something called plasmodium. Okay. And they're named for a structure called an apicoplast. Apicoplast. An apicoplast is a non-photosynthetic plastic. I'm just going to reduce it. They're also known for something called sporozoites. These are infectious cells. And these are really neat. You remember I said we're going to only do a few life cycles? Yeah, we're going to do plasmodium. Okay, so plasmodium is really kind of neat. All right, so we're going to start at part 1. I'm going to have to do a lot of talking rather than writing on this. You may find this as an essay question. Perhaps.
Figures in the book, correct?
Oh, yeah. It's big.
Isn't this one 8.10?
Should be. Okay. All right, so it starts here. This portion, human. This portion, mosquito, Anopheles. Okay, the Anopheles mosquito. So what happens? The Anopheles mosquito is infected with sporozoites. Okay? All right, so what happens is this infected mosquito bites a human. And it transfers the sporozoites into the human. And the sporozoites actually migrate to the liver. The sporozoites migrate and move to the liver. And they infect a liver cell. And in the liver cell, they become what we call merozoites. Okay, so in the liver cell, they become merozoites. So a different type of cell. Form inside the cell, in the liver cell. And then they break open the liver cell and get out into the bloodstream. Now, apicomplexa, one of the things that they're known for is that they have this apex right here. See how it dips down like that? Right there? It literally takes that apex and drills into the red blood cell. Also known as an apical complex, but it takes the apex of the cell and drills into the red blood cell.
That's a merozoite?
Yeah, the merozoite. Yeah, a sporozoite is an infectious cell, so meaning that it goes from that one organism, the mosquito, and infects the human. But once you get into the human, it transforms, it changes into a different type of cell, and that's the merozoite. And the merozoite actually infects red blood cells. And what happens is when they're in the red blood cell, they burst through the cell. This causes fevers and chills, okay? And they continue to infect other red blood cells. So any questions so far? Yeah. I don't care, whoever. Whomever.
So merozoites, how do they transform? How do they just like, something happens in the liver, they just change?
It's not actually in the liver. What's happening, I'm sorry, it's not actually in the red blood cell. What's happening is they're going from these amoeboid types. I haven't used that term yet, but amoeboid, they're like small cells that move. Okay, they go from these small amoeboid sporozoites, which are moving around, into these more stationary merozoites that have a drilling portion. So they convert, they change. They actually change form. Yeah, they divide, go through division.
Does that only happen in humans, or? I mean,
Yeah, no, it's a two-host parasite. Yeah. So what happens is it has to have a mosquito. It has to have a human. So
it won't work on like animals. Like if the
not typically. This is malaria. The Plasmodium causes malaria. Yeah, it's a two-host organism. All right. So what occurs? Inside the red blood cell, they form gametocytes. So these are cells formed in the red blood cell, the gametocyte. Everything so far of the Plasmodium is haploid, only has one complement. Okay, that's what the blue means. Everything so far. So what happens? Well, another mosquito bites an infected human and picks up the gametocytes. The rest of the development of the apicomplexan, Plasmodium, occurs in the mosquito. So the gametocytes produce two different types of gametes, male and female. The female are these kind of roundish gametes. These are the male, not this. This is gametocyte, this yellow one. That's the gametocyte cell. This is the gamete. They're haploid. Right? So this is haploid, and this is haploid. They fuse, which is what's happening here, fertilization. Fuse the gametes. You get a zygote. N. Any questions so far? This is in the mosquito. Yeah. It's actually occurring in the gut of the mosquito. Sorry.
The mosquito carries the gamete.
Yes. Yeah. So what happens is a mosquito bites a human. It injects into the human these infectious cells, sporozoites. The infectious cell migrates to a liver, changes into a merozoite. The merozoite gets into the red blood cell, produces gametocytes, which is another cell. Those are then picked up by the mosquito.
I'm sorry, like the second mosquito, you're saying it by so it must already carry one out.
No.
So,
The gametocytes, these cells, can produce both types of gametes. Yeah.
Just picking it up from the human?
From the human. Yeah, so it's picking up the gametocyte from the human. And you get the formation of both types of gametes, male and female. Okay? Anything else? These gametocytes aren't singular in one human. So a human's not going to have only male gametocytes. It's going to have a whole bunch of them, and they're going to form a whole bunch of different types of
How did you
gametes.
get the zygote in the human body and you only get it in the mosquito?
What? Where's the zygote in the human body?
Yeah, why doesn't it form in the human?
Because it doesn't. It needs the mosquito digestive tract. It just doesn't. These don't form gametes inside, the gametocytes don't form gametes inside the human. It has to have the mosquito to form the gametes. And in order to get a zygote, you have to fuse gametes. Does everybody follow that? No? Is everybody, any questions? Yeah.
So this portion that's happening, the fertilization of meiosis, that's happening in another mosquito that
Yes, different mosquito. Yeah. Anything else? Okay. So you get the formation of a zygote. This is inside the intestine of the mosquito. Actually, the gut. Let's just call it the gut for the mosquito. It migrates to the wall, to that gut, and it forms something called an oocyst. An oocyst. So the zygote goes through meiosis and forms this protective structure called an oocyst. Inside the protective structure are haploid sporozoites. How did we go from a diploid zygote to a haploid sporozoite? Yeah, it's right here, meiosis. So that's what happens. We get the formation of a zygote, right here. It's occurring inside the gut. Inside that gut, inside the zygote is surrounded by a protective layer. It becomes what we call an oocyst on the lining of the gut. And inside it goes through meiosis to produce sporozoites. What are sporozoites? Infectious cells. So look, you end up with sporozoites. These migrate to the salivary glands of the mosquito. And then the mosquito bites another human and it continues again. Any questions? Yes.
Can you hear about the oocyte?
The oocyte, what happens is once you get the formation of the zygote, it produces this protective structure around it. And so the zygote goes through meiosis and produces this protective encasing. And when it goes through meiosis, inside this encasing, that whole thing with the encasing and the cells inside is called an oocyte. When it goes through meiosis, it produces sporozoites inside. Then it bursts open and releases these into the mosquito, which then migrate up into the salivary glands, bites another human, and starts it again.
Is it possible that mosquito infect another mosquito or
Another mosquito? No. No. One mosquito is not going to infect another by biting, and these migrate to the salivary glands. I don't know how it would if it occurs, because if it does, I have no clue how it would do that, because they don't bite each other. Yeah. And the only way you're going to infect it is they're in the salivary glands, and when they bite a human, they inject those sporozoites into the human. And as for cross species, that I'm not positive, but I don't think that occurs. This is pretty much mosquito-human. Anything else? No, what? Well, actually, can you? Yeah, how about after? Okay. All right, ciliophora. Ciliophora. These use structures, small hair-like structures called cilia, for feeding and movement. Hair-like structures on the surface for feeding and movement. All right.
Can you go back to the foot?
Feeding and movement. Cilia. Hair-like structures on the surface of the cell used for feeding and movement. Okay. Now, one of the things about cilia is they have what we call a large macronucleus and a small micronucleus. The macronucleus is used by the cell for everyday function. The micronucleus is typically used for reproduction. All right, so let's look at paramecium, which is an example. Paramecium. All right, so let me identify some of the structures. This is a contractile vacuole. It has these radial canals. And what happens are these radial canals take in excess water from the cell. They put it into the contractile vacuole, which ejects water from the cell to prevent too much water getting into there and causing problems like exploding, okay, breaking open. So what happens? Well, one of the characteristics of a paramecium is this structure here called an oral groove. The oral groove is lined by cilia, and the cilia beat in food materials, a lot of bacteria. And they move this food material down this groove to the mouth. Called the cell mouth right there. And in the cell mouth, or at the end of the cell mouth, the paramecium develops what we call a food vacuole. And the food vacuole contains whatever the paramecium consumed. And we didn't talk about these, but there are lysosomes inside the cell. They contain digestive enzymes. Don't have to know what a lysosome is, but they contain digestive enzymes. And so what happens is a food vacuole will actually merge with a lysosome, and it breaks up the material inside it as it moves around the cell. Okay? And whatever's left, whatever waste product it is, it ejects from the cell. That's what these little things here is waste. Okay? Now, this is a large macronucleus and a small micronucleus. And the little hair-like structures right there are cilia. So that's the general structure of a paramecium. On the exam? Yeah, you gotta know paramecium. Oh yeah, you gotta know euglena. Nothing else, you gotta know those two. Okay, those are two big ones. You probably have those on your lab practical too, won't you? You gotta saw them in lab, so this is all just review for you. Yeah? No? Kind of? Okay. Every week we're trying to cover all of it. So, life cycle of paramecium? No. We're not going to do it. It's incredibly complicated. I could go over it, but I don't have time. So if you're interested, you can come up and ask me. I'll go over it. It's very complicated. It has to do with different mating types, merging, and the micronucleus going through divisions and then becoming macronucleus and going through binary fission. Even I wouldn't give you that as an essay. Okay? So it's very interesting. It's in your book. You can read on it and come ask me about it. That's fine. But I'm not going to do it for lecture. I just don't have time. It takes quite a while. All right. Any questions? Nothing? All right. Stramenopelia, that's another kingdom. These are characterized by having both a smooth and hairy flagellum. All right, let's go through some of the phyla. Right. The diatoms. These have shells or casings that are made of silica. What else is made of silica? Glass. They're actually glass. So they have these glass casings. They're absolutely gorgeous. They're beautiful proteins. And they have the tests, the shells, actually have little holes through them. And if you look, they kind of have this shape to them. And they're responsible for diatomaceous earth. Okay, so they're very important in diatomaceous. Right? If you look at a picture of them, look like this. One example anyway. Look at how beautiful these are. They're all different shapes. I don't even know if this is the one that came out of your book. If you look, yeah, they're all different shapes. They have these two shells. Okay, they have an upper covering, and then they have a lower covering. And they're made of this very fragile material, this silica, and it has all these little pores. You can actually see the pores. Look at all the little holes right there. Okay, and the flagella coming out. They're absolutely gorgeous. Okay, and what happens is they'll die, and they'll sink to the bottom of the sea. And one of the things they do is they take carbon dioxide out of the air. We're really good at putting carbon dioxide into the air, which increases the amount of material being held in the atmosphere. And so basically it's like wearing a really heavy coat. And so we're increasing that temperature. And one of the suggestions, yeah, it's one of those, I'm not sure how much I would agree with it, but I'm not sure I wouldn't. I don't know yet. Is to increase blooms of diatoms to remove carbon dioxide out of the air to reduce the amount of pollution that we're putting in. Problem is what would that do, right? So the thought is you put a whole bunch of carbon dioxide in the air, these take a whole bunch of carbon dioxide out, they die, they sink to the ground, to the bottom of the ocean, they have that carbon dioxide in their bodies, it's now removed from the atmosphere, reduce the environment. That's another change. Okay, so when we do ecology, that could be a real big problem. We'll actually talk about that. Okay. All right, Chrysophyta, which are golden algae. Right here, Chrysophyta, the golden algae. These have brown yellow pigments. Brown yellow pigments. And they also have something called biflagellated cells. Now, most of the golden algae are unicellular. But some are colonial. And this would be a colonial example. If you look, didn't come out real well, but there are little flagella here. Then there's this outer protective coating. And the actual cells are here. Any questions on that? Again, do you have to draw this? Why did I do this? And please believe me, this took me a long time to do. We're talking a long time to do. Why did I do this? Yeah, just so it's easier for you. That's it, okay? So what I would do is the material I'm putting on the Elmo, make sure you get it on here. This is a single page that's very easy to walk around with, right? Not so easy to walk around with a stack of material, but if you're going to be sitting in line somewhere, it's easy just to have a quick check, right? That's what this is. It's not everything, but it's a little bit easier to keep things related. Okay, Phaeophyta, brown algae. These are multicellular. They have something called a thallus. And a thallus is the body structure of a kelp. Kelps are not plants. These big, big photosynthetic organisms that make up kelp beds are actually, most of them are these brown algae. And if you look at them, they have several structures to the thallus. They have the blade, which are these right here. These leaf-like structures. They're not leaves, but leaf-like. They're up at the top. Little leaf-like structures are the blades. They then have this thing called the stipe. It's actually for support. So brown algae don't have what we consider roots. They have a structure called a holdfast. And so the holdfast holds down on the ground. It's named for what it does. It holds fast to the ground. But it doesn't absorb nutrients. It doesn't take up nutrients through that structure. And then coming off the structure is this stipe, or this support system. And what it does is it supports the blades so that they can get them closer to the light so they can do photosynthesis. And this is production of food. They don't have true roots. They don't have what we call true stems. Very well adapted for water environments. Their consistency of the cell wall allows them to move. So if you think of a pine tree, probably not going to move too much if you put it in wave. But if you think of, if you've ever gone, well, you guys will be doing a marine field trip. Have you guys done that yet? No? Okay. Well, when you go, you'll probably see some of these, or you've been to the beach, you've seen these kelps, right? They're a little bit more fluid, a little bit more moving. Well, you need that, especially in water environments. They don't have to support their weight because that's what the water does. So they don't have to pull against gravity. They're able to be supported by the water. And so they can get very, very large without this very heavy material, very strong material in the cell. Okay. Any questions? Everybody's just kind of going, oh my God, what?
Is the base alive or dead?
I think it's alive. I've never heard of it being dead, but I don't actually know. You've got to feed nutrients down there. So I've never heard of it being actually dead. That would actually be a great question for Dr. Kovnat because he's a marine biologist. Any other questions? No? Everybody okay? Thursday, waiting for the weekend? It's going to be all biology, right? Lie to me and say yes. Okay. Good. Because I'm going to be writing part of the exam today, this weekend. So make sure you guys study. Yeah.
When will we have the essay questions?
Oh, I don't know that I'm actually giving you essay questions. You guys want me to give you the essay questions? Well, I gave you one, right? Life cycle of Plasmodium. Explain that. You got one, huh?
So Sean study that one?
So far. So far, I haven't come up with them. I'll try to come up with them this weekend. I haven't done the other life cycles, so the other life cycles are going to play a part in that. We'll get to it. Okay, my coda. These actually used to be put in with fungi, and there's different reasons. They're similar to fungi in the fact that they have hyphae. Oh, speaking of this, We won't be doing this life cycle either. The, no, we won't be doing the brown algae life cycle. Okay, so similar to fungi in the fact that they have hyphae. Hyphae are these filamentous strands of material. They typically have lots of nuclei, so they've gone through lots of mitotic divisions without actually dividing their cytoplasm. Got lots of nuclei. And they're used for absorbing material because they have a very large surface area. They tend to be thin but very long, and so they have this immense amount of surface area so they can uptake material from their environment. Hold on a second. All right. Another way that they are not, another way that they're not like fungi, so this is how they are like fungi. And the way that they're not like fungi, they have flagellated cells in their life cycle. Fungi do not. They're similar to fungi in the fact that they have no plastids. They are not photosynthetic. They don't have plastids. But they're different from fungi. Their cell walls do not contain cellulose. Actually, they have a structure called chitin. The same type of material that's in the exoskeleton of an insect. It makes the insect go crunch when you step on it. That's the chitin. Okay, so the fungi have this chitin in the egg. Yeah, it doesn't sound very pleasant, but that's what it is. That is a branching material of glucose that's actually in the walls. So the water moles have cellulose. Oops, I'm sorry. They have cellulose. They do not have chitin. Okay. Fungi, true fungi have chitin, and water moles have cellulose, not chitin. All right? And this is the life cycle of a water mole. And you don't have to know it. I won't ask you that one. I promised you two. I will do two. We did plasmodium. Any questions? Nothing? Okay, cool. Rhizaria. Rhizaria are amoeboid type of organisms. We're not going to do cercozoans. Okay, so I'm not even going to divide those out. I'm not going to even talk about them. I'm going to talk about foraminifera and radiolata. Foraminifera are amoeboid. Have you guys seen amoeba?
Yeah.
Oh, good. That has got to be one of my favorite proteins. Did you guys see a living amoeba? Okay. Okay. So amoeba, but these have these kind of thread-like pseudopodia. You guys know what pseudopodia are, right? False feet. When you saw the amoeba, looked something however it was, and it's got this thing. These are called pseudopodia. Podia pod refers to foot. Pseudo means to false, so they're false feet. Did you guys watch it eating? Did you get to, no? Okay, sometimes you do, and sometimes you don't. They actually use this to extend out and feed. So what happens is they extend these out. Did you watch cytoplasmic streaming?
Yeah.
Yeah? Okay. So what happens is they liquefy the portion here, and they push the plasma membrane out, so they move this way, and they solidify this portion of the cell. And then this is what we typically think of for an amoeba. But The foraminiferous and radiolarans don't have these type of pseudopodia. What they have are these really long thread-like pseudopodia. And if you're a radiolaran, they radiate out from this big center. Okay? Both of them have these shells or tests. So foraminifera have these thread-like pseudopodia that come out of a porous test. Porous just means holes. And the shell is calcium carbonate.
What shell?
I'll show you in a second.
No, what is it written? What's written?
Porous shell. Okay? All right, so if you look at the foraminifera, it looks like this. Look, here's this amoeboid, and it has these little pseudopodia that come out of this shell that protect it. Did I mention that amoebas, I just love amoebas? Hint, hint. Good hint, right? You're going to see amoeba on the exam. They're just too cool not to put on. They're really, you guys saw them in lab. You can't tell me those weren't neat. They weren't interesting. They weren't easy to remember. Yeah? Okay, well, there's different kinds. All right. Radiolarans, look at this one. Okay, so look at the difference. You see in the radiolaran, you have, look at yours. You've got this circular area here, this test around it, and they all radiate from, these pseudopodia all radiate from the general same area. The circle, the center, radiolarian, they radiate out. Okay? The foraminifera have these porous shells, okay, called tests. And you've got this huge structure, and then see how you have a whole bunch of them all over? They're going through the holes in the pore. So you see the difference? These have them, they're kind of, not equal because they're not always the same. But think of a tire. Think of a hubcap on a tire. That's the radiolaran. It looks a little bit more like a hubcap, the spokes coming out on the hubcap. In a foraminifera, they don't. There's just a mass of pseudopodia. Okay. All right, so let's do radiolara. These have thread-like pseudopodia that emerge or radiate from a central body. So how would you go about memorizing all this stuff? A lot here, isn't it? Or is it just me that thinks there's a lot here? There's a lot here, right? You're majors, so I expect you to know it. But rewrite it. Make sure you can redo this. You're not going to have to draw it on the exam, but if you can rewrite or at least spell it or identify the structures, that's what you want to do. You want to be able to draw this. You want to be able to write it. You want to be able to discuss it. Perhaps get into study groups. Okay, go. Archaeoplastina. These are closest relatives to land plants. Okay. All right, there's only two that we're going to actually go through today. Look at Rhodophyta, which is this upper one, the red algae. Okay, it's identified by its pigment. Phycoerythrin. Gets its name because the phycoerythrin masks the green chlorophyll. The other two groupings contain plant-like chloroplasts. All right. Rhodophyta, if you look at it here, okay, it's kind of reddish. I saw it very well. These reddish, red algae, these structures, okay? They're actually very beautiful. Some of them can be kind of feathery, which is what this is. This little feathery type of structure. Some can be these little blades. You'll actually see these. You probably will see these, especially this kind, when you do your marine. This kind of flat blade. They are in the tide pools. And we're not doing any life cycle for them. Green algae. Two groupings, Chlorophyta and Charophyta. Charophyta we're not covering here. Charophyta we're not doing here. Chlorophyta, we have to. Okay. Most of it I have down here for you because I knew I would be running out of time by this time. All right, so they're named for their grass green chloroplasts. That's what gives them these green colors, the grass green chloroplasts. They're very similar to land plants. It used to be when I was taking this class, we used to say land plants evolved from ancestor of Chlorophyta. Now it's more likely that they evolved from ancestor of Charophyta. Okay, that's why we cover Charophyta when we get to the plants. We don't cover it here. Okay. You have different numbers of cells of Chlorophyta, or I'm sorry, the green algae themselves. You can have unicellular, one cell, Chlamydomonas. Colonial volvox, that's what this one is. Okay, this is Chlamydomonas, this is volvox. This is ulva. This sea lettuce, I think, is the common name for ulva. And then multicellular, which would be ulva. Yes, you have to know. I make my Bio 3 students know, and you guys definitely need to know. Okay? All right. Complex life cycles, Chlamydomonas. You'll be happy to know that because I ran out of time, I'm not going through it. So you don't have to know it. But wow, is it a neat go through it. You have different mating types. They actually merge together. One of the things you should realize is that a lot of protists do what's called asexual reproduction. So a lot of them will go through asexual reproduction when times are good. But when environment goes bad, they'll go through sexual reproduction so that they can recombine the genes. And so perhaps get a organism that's better able to combat whatever environmental issues are going on at that time. So a lot of times sexual reproduction, especially in these circumstances, we'll get to some of that with fungi too, occurs due to environmental conditions going bad for the organism itself. Okay. Charophyta and Plantae aren't covered here. All right. So does that mean that you have to know Charophyta and land plants? These are Plantae. No, you don't have to know them for the exam. So there's a couple you don't have to know. You have to know them for the next time, so don't cross them out. All right. Unicons, these are the closest ones to fungi and animals. Do you guys remember when we were doing kingdoms and I was talking about eukarya, the domain eukarya, and I said fungi are actually more closely related to animals than they are to plants. Okay, well, they are. They're more closely related to animals than they are to plants. They're put in the supergroup Uniconta. Okay, and it's very diverse. I mean, think about it. Fungi, mushrooms, the mushrooms you put on your salad, and humans. Very diverse, right? And everything in between them. All right, so they're more closely related, considering. Amoebozoans, that's the only one we're going to go through, right, are the amoebozoans. These have an amoeboid body. The chloroplast in the amoeba is this kind of, I'm sorry, not the chloroplast. The pseudopodia are these kind of lobe shapes. This is probably what you looked at when you were looking at amoebas, these lobe shapes, these kind of things, lobes, big structures. Yeah, that's usually what you look at. All right, so Myxogastridia and Dictyotellida, sorry, it's a new one for me, is going to have fruiting bodies. I just remember them as cellular slime molds. They didn't have them classified out when we were doing it. Okay? This is the neat thing about them. The plasmodial slime molds, the feeding structure is called a plasmodium. The cellular slime molds, the feeding structure, is a cell. That's how they differ. One is this huge plasmodium, and one is a simple little cell. Gym amoebas, these are unicellular and heterotrophic. And entamoebas are parasitic. Okay. All right. The plasmodial slime mold. The Myxogastrida is the plasmodial slime mold. This one you need to know, only because it's just really interesting. They produce what we call a feeding plasmodium. It's basically a supercell. Sure. It's basically a supercell. And what happens is it moves and consumes organism as it does so. When the plasmodium matures, it becomes this kind of web-like material. And in harsh conditions, the mature plasmodium starts to produce these fruiting bodies called sporangia. This is a young one. This is maturing. Okay, so sporangium is a fruiting body produced by the plasmodium. And what happens is you get, here's a mature sporangium. What happens is this body produces this stalk that grows up and elongates, and you end up getting this mature sporangium. Inside the mature sporangium are cells that go through meiosis. So these cells go through meiosis. When they go through meiosis, they reduce the number of chromosomes and they become spores. So cells in the mature sporangium divide by meiosis and produce spores. That's what these are. These are haploid. Now these spores are released by the sporangium. They end up going to lots of different environments, and they germinate. Did you guys do what germination is? To grow? So fertilization is getting gametes together. When something germinates, it breaks out and it's going to start growing. That's what we're talking about here with the spores. So when they get to an environment that's conducive to their structure or their life cycle, so in a favorable environment, the spore will germinate. Because they break out of the spore, they start to grow, and they produce different types of cells. They produce what we call flagellated cells and amoeboid cells. Flagellated have flagella, amoeboids have the amoeba shape. But what happens is these cells pair up in like kinds. So similar cells pair and form a zygote. Because see, each one of these are haploid. So they pair and form a zygote. That's called fertilization. The zygote becomes a young plasmodium. And the cycle starts all over again. Okay, so let's go over it real quickly. Feeding plasmodium. Kind of like a supercell. They're really big, lots of nuclei. It moves. It's really kind of neat, but it moves very slowly, so you can't really see it per se. This plasmodium matures. It becomes kind of this web-like structure, web-like plasmodium, that starts producing fruiting bodies, which is right here. Those fruiting bodies are called sporangia. Sporangium is singular. The sporangium inside it has cells. Okay? So as it matures, it has these cells that go through meiosis. They become haploid, and they become spores. The spores are released by the sporangium. They're released by the sporangium and end up in some favorable environment, whatever it might be. The cell breaks out of the spore, and there's two different types. There's some that are flagellated, there's some that are amoeboid. Two amoeboids get together, or two flagellated get together to produce a zygote, and then it starts all over again, and all over again.
What happens with the stalk?
Oh, it's just nothing. No. It just disintegrates. Not a big deal.
So the flagellate and amoeboid don't get together?
No. Yeah, believe it or not, no, they don't. They get together as like pairs. Anything else?
So for the left side,
Anything else?
There's just different cells.
Yeah, they produce different ones. They come out of the spore, yeah, as different types. The dictyostelidia are the cellular slime molds. I don't have time for that. We talked about gymnamoebas, entamoebas, real basically, but you have got to see these. Look at this. All right. The cellular slime mold, the feeding structure. In the plasmodium slime mold, you get this huge plasmodium that feeds. That's what moves around. In a cellular slime mold, it's a single cell. That's the feeding structure. But look at this. This is actually an aggregate. So when things are having problems, when the cell needs to move from one place to another, It's not so easy to move as a cell. So what happens is a whole bunch of these cells get together and form this huge aggregate. Now it's not like this, it's small, but this big cell, it's kind of like a supercell, that actually migrates. When it gets to a favorable environment, it produces a stalk and then it goes through its life cycle. Look at that, that's an actual cell. Actually, it's a group of cells that have come together to move from an area that's not favorable to an area that is favorable, so that they could generate more organisms. Yeah, it actually moves like an amoeba. It has that kind of amoeboid movement. It's really kind of neat. The best description of it I can think of is snot. It kind of looks like snot. Good description, though. It is. It's kind of glistening, and I'm not done yet. So sorry, guys. You're going to have to wait. I've got to finish this today. Otherwise, fungi will be scary because I'll have to finish it. Okay. We're not going to do animal and fungi. Don't have to. I'll get to it. Ecological roles. Protists can be symbionts, which means they can live with other organisms. Okay. They can be parasites like Plasmodium, Listeria, which actually kills fish. Okay. They can digest cellulose. Termites. Termites don't digest cellulose. Plants. So when they eat our houses and stuff, they're not digesting anything. Here's the protease in. They're also photosynthetic, so they can produce. They're producers. They produce material. Photosynthesis. And then you can go ahead and take a look at this. This is a food web. Get used to seeing this, okay? Because this is going to come up in ecology. I better warn you, I'm an ecologist, so I like ecology a lot. All right, so we're going to be actually doing food webs. No, you do not have to memorize it. This is what it is. Arrows, may as well tell you now, arrows tell you the direction energy is moving. Basically food. Okay? So producers, small organisms, are eaten by larger, or the larger, like the penguin here. Okay? If you had the arrow going the opposite way, you'd be saying the protist is eating the bird. Okay? Unless the bird's dead, that's probably not happening. Okay? So the arrow, the direction of the arrow, this tells you energy is moving that way. Alright? So you may as well get used to it because we'll actually see it a lot. Okay? But I'm not going to have you put that on there. You just need to know it. This table is in the back of your book. Not the back, I'm sorry, the back of the chapter. This is what I put on that flow chart. This information is on that flow chart I drew for you. Okay? And I took this different information. The examples you need are any examples in this table. Those are the only ones I'm going to ask you. Anything else? That's it. Okay, so those are the only things I'll ask. Does anybody remember how I said I'd ask you the names? Matching. It's matching. So I might put Giardia. You tell me what it is. Okay, it's not fill in the blanks, but it is matching, so you do have to know. All right. Have a wonderful weekend, guys. We'll do